Good evening. Welcome to the 2025 University of Georgia 3-Minute Thesis Competition. Yes. My name is Meredith Welch Devine. I'm the Assistant Dean of the Grad School, and I'm absolutely delighted to have you here tonight. Now more than ever, it's critical that the general public understand how universities make their lives better. And a huge, huge part of that is our scholarship and the research that we do. But they won't understand the impact that we have in their communities unless we tell them. And unless we tell them in a way that's exciting and engaging and understandable. And that is what 3MT is all about. So the students that you'll see tonight are to be commended not only for their excellent research, but also for that dedication to ensuring that their work makes it beyond the walls of the ivory tower. And of course, for their bravery. This is going to be a really stellar show, so without further ado, I would like to introduce your MC for the evening, Mr. Nick Laconico.
Thank you so much, Meredith. Good evening, everyone. My name is Nick Laconico, and I serve as an Associate Director for Business Engagement at the University of Georgia. To make the most of our evening, we have a few informational slides to go through, but we want to engage you. Our presenters have worked incredibly hard over the past couple of months, honestly, years and probably their lifetime, to get and share this information with you all. So we want to feed off your energy. We want you to bring that energy, bring that excitement, because they feed off of you. So to engage you a little bit, I'm going to ask you to do something. So I raise my left hand. I'm going to ask you to say, ooh. Yes. My right hand, say, ah. Yes. And both hands are going to clap. Yes. Okay, great. So I need you to bring that energy for these instructions and this rest of the night so our presenters feel that. So the 3-Minute Thesis is an international research communication competition, better known as the acronym 3MT. The competition requires doctoral and master's students to explain their thesis or dissertation topic and its significance in 3 minutes or less using only a single static presentation slide to a non-specialist audience. Ooh, there you go. Started by the University of Queensland in 2008, 3MT is now a globally recognized competition with over 900 plus institutions across 85 countries holding their own events. Ah, yes, there you go. The University of Georgia added its name to that list in 2012, and it has grown since then, now occurring twice a year and engaging both the UGA and Athens-Clarke County communities. Presenters must use only one static slide without any additional electronic media or props. Presentations must be spoke and not exceed 3 minutes, with time starting when presenters begin speaking or moving. Lastly, the decision of the judges panel is final. Ooh. While all our presenters are already grand prize winners, in our eyes, their presentations will be judged by the following 6 areas. Presentation provided clear motivation, background, and significance to research question. Presentation clearly described the research strategy design and the results findings of the research. Presentation clearly described the conclusions, outcomes, and impact of the research. Ah, here we go. The oration was delivered clearly, and the language was appropriate for a non-specialist audience. The slide was well-defined and enhanced the presentation. And lastly, the presenter conveyed enthusiasm for the research and captures and maintained the audience's attention. Here we go. So we have 3 awards, and each award winner will receive a plaque. The People's Choice Award winner will receive $500. Ooh. The runner-up will receive $750. Ah. And the grand prize winner will receive $1,000. You got it. And now I'd like to introduce our judges. First, we have Mr. Jeff Montgomery. Jeff Montgomery moved to Athens in 1995 after graduating from Furman University. He joined the Athens-Clarke County Unified Government's Public Information Office, now Communications Department, in 1998 and has led it since 2013. The department communicates information about ACC Gov's programs, services, and initiatives in digital, print, and video formats. Manages, distributes networks such as accgov.com, the ACTV TV channel, digital signage, streaming service channels, and social media outlets, and engages with media, residents, and the community. Jeff also serves as the local production liaison by operating the Athens Film Office. A graduate of Leadership Athens and a certified public communicator program at TCU, he has been active in boards and steering committees over the years for the City Council Communications and Marketing Association, 3CMA, Community Connections of Northeast Georgia, Youth Leadership Athens, the Convention and Visitors Bureau, Boy Scout Troop 1, Cub Scout Pack 1, and AthFest. Ooh. Next up, we have Miss Dominique Holloman. Dominique I.H. Holloman, a UGA alumna of 2001 and 2004, works as a government and public affairs executive specializing in government relations, strategic advocacy, and legislative engagement. Over the course of her career, in addition to government and politics, she has worked in the sports industry and nonprofit sector. Her career trajectory has given her extensive expertise in policy analysis, advocacy, relationship management, and strategic communication. Dominique holds a Juris Doctor, a Master of Education in Sport Management, and bachelor's degrees in sociology and psychology from the University of Georgia. A volunteer leader for UGA since 2007, she was the inaugural president of UGA Black Alumni Leadership Council. She additionally is a past member of the Law School Alumni Council and an emeritus member of the UGA Board of Visitors. Most recently, she served on the executive committee of the UGA Alumni Association Board of Directors and currently sits on the Graduate Education Advancement Board. She is a member of the 2017 class of UGA 40 Under 40, the 2018 recipient of the Nyota Tucker Trailblazer Award, 2018 convocation speaker for the Mary Frances Early College of Education, and a 2022 and 2023 Master of Ceremonies for TEDx UGA. There we go. And last but not least, we have Dr. Kostas Spiro, who serves as the Chief Innovation and Entrepreneurial Strategies Officer for the University System of Georgia, also known as USG. In this role, he focuses on developing and implementing strategies to advance innovation and entrepreneurship in alignment with USG's mission across research, education, and public service. He leads efforts to create shared programs, startups, incubators, and funding mechanisms that enhance USG's collective impact and support the state's economic development goals, workforce needs, and technology transfer initiatives. Spiro also holds a faculty position in the Ivan Allen College of Liberal Arts at the Georgia Institute of Technology. Let's give it up for our judges. And so throughout the program, we want you to keep track of the presenters and your favorite presentations, because you all will vote on the People's Choice Award. So feel free to jot notes down on your program or elsewhere so you can make sure you vote at the end. So a few other housekeeping tools. We do respectfully ask that you silence your cell phones, so put them on Do Not Disturb, turn the volume down so that we can give our full attention to our presenters. But as I said, you'll see it on the screen coming up soon. We will be engaging you throughout the presentation with each presenter through a medium called Menti. And you'll see that on the screen. You can scan the QR code in a little bit or go to menti.com, and we will actually ask you to engage in that polling throughout the session. And so you can see there, you can open a browser and go to menti.com, or scan that QR code, enter that code, and you can keep that up throughout the whole presentation tonight, and you'll be able to engage with our panelists, or with our presenters. They have some fun facts you'll learn about them, and just really engage throughout the presentation tonight. So to give this a test, here's the first question. How are we all feeling? So, it's Thursday night. We're super pumped. We have a great presentation out. So we have some people online too. So thank you for going ahead and responding there. We're feeling excited. Maybe some internal screaming. So go ahead and fill that out. We're happy no one's sad. But great. You're all doing great. So let's give it up for our first presenter, right? We have Cameron Wysocki, studying psychology and vision science. His presentation is Eat Your Greens, How Plant Pigments Help Us See Farther.
What's your favorite type of pizza? Can you just shout it out for me? Got a lot of cheese. Pretty good choice. Garbage? My favorite type of pizza is spinach. I love spinach. I eat almost every day. I put it in pasta, I put it in soups, I put it in stir fries. If I can find an excuse to eat spinach, I will. And it's driving my girlfriend a little crazy. Now I want to share with you all one of the reasons that I love spinach so much, which is that it's likely helping my vision. When I eat spinach, or any other leafy green vegetable for that matter, two antioxidant pigments, lutein and zeaxanthin, are going to begin to accumulate in my eye. In the eye, they filter blue light, just like blue light filtering glasses. One important functional consequence of these pigments accumulating in the eye and filtering blue light is they might help us see farther in the distance. Now, blue light is going to be scattered by the atmosphere more than other types of light. This is what makes the sky blue. Now, all this scattered blue light is also going to cover up faraway objects, making them blurrier, hazier, and harder to see. Eventually, it's going to help determine how far away something is before we lose sight of that. You can see that demonstrated behind me. This airplane right here is covered by more of this atmospheric blue light. You're probably going to lose sight of it before the other airplane. Now, you might be thinking, we have these plant pigments in our eyes. They help us see farther in the distance. They are filtering blue light, so they might help us see farther in the distance. So for the first phase of my project, I tested this. I brought people into the laboratory. I measured the amount of lutein and zeaxanthin in their eyes, and then I simulated their ability to see in the distance by presenting them with targets that were covered with blue light that matched what you'd find in the atmosphere. For the next phase of this project, I plan to bring people into the environment, measure the amount of these plant pigments in their eyes, and then evaluate how far they can actually see in the distance. This research has functional importance for individuals whose jobs and occupations involve seeing things far away, such as pilots, air traffic controllers, truck drivers, and professional athletes like baseball players. But I think it's also important for individuals like myself and some of you in the crowd who just want to maximize their vision. My study and my results suggest a simple dietary intervention to be able to do so. That's eating more leafy green vegetables. For me, though, that just means continuing to eat more spinach. Thank you. I tripped up.
Awesome. We'll give it up for Cameron. So, as you can see, you will be seeing a question after every presenter. And so now getting to know Cameron more, he was asked this question, and you're guessing his response. Our folks online are already ahead of the game. So on your phones, you should have this question and be able to submit a response. It will give you a few moments to answer. And so while you're thinking about that, think about how you can include spinach now, right, in your next meal. Maybe a pizza, a salad. If you're still hungry after this, there's some great restaurants downtown, but really thinking about it quietly. Or not. Sorry, that's just my personal wanting to do that. Power, right? No, but we're seeing Popeye, which makes sense. I thought about Popeye while Cameron was speaking. And so we're super excited to have this presentation, each one. So as you see the presenters leave, pull your phone up, look at that question, and we will make sure that you are kind of guessing. They're not all about your project mascot. We have some other fun ones, some memes, some other questions, but we are really excited to learn more about our presenters through that. You all should have also received a card when you came in, so kind of reflecting on the presenters coming up. Right, remember, we're still wanting you to vote on people's choice at the end. So think about the things Cameron said, write some notes down, because if he is your people's choice, you want to make sure you remember what we talked about and vote for him at the end of the presentation after hearing all great 10 presenters here. I love the discussion happening. It's going to be great. So no one thinks Paul Bunyan. Okay, that's fine. That's fine. But we are having our great judges think through all the great things Cameron said. And so we're giving them some time to judge and to score. And so our last MC sang, but you all don't really want to hear me sing, so you can offer that maybe? like a sad, but it's all good. We're going to make sure we keep the show going and running, and you'll hear our great presenters coming up soon. All right, so I think we have our next presenter ready. Next up, we have Hadley Copeland, studying wildlife sciences in the Warrenell School of Forestry and Natural Resources. Her presentation is entitled Riss Runs Downstream, Managing Forested Watersheds.
Take a deep breath and really fill your lungs. Now, for my friend Leonard here, breathing looks a little bit different. Leonard is a lungless salamander, meaning he breathes exclusively from his moist skin. And in the Appalachian Mountains, that moisture comes from the shade of hemlock trees that keep the forest cool and damp. However, those hemlocks are under attack by an invasive insect. And as the canopy disappears, forests grow hotter and drier, putting salamanders like Leonard at risk. To combat this, forest managers are applying a special pesticide. And while this protects their habitat, we don't really know what it's doing to the salamanders downstream. My research reveals those effects and helps us make better decisions for our forests. But first, why do we even care about salamanders in the first place? Lenny's cute, yeah, but odds are he's going to get eaten by the next big thing that comes along. And that's exactly the point. Salamanders are able to eat small insects that feed on plants and decaying matter, take those nutrients up the food chain to birds, reptiles, and fish. In doing so, they promote healthy forests and clean waters. To see if this is something that is affected by pesticides, I start small, and I look at the brains to see if regions like the cerebellum are affected by this pesticide, which has already been seen in other species. I then look under the microscope to see how those structures are affected, and then connect that to behavior to see if they can still eat or hide from predators effectively. Finally, I scale up. I found that adult stream salamanders are already impacted by these pesticides in these forest systems, and that shows that long-term effects do have an impact. Connecting behavior to brain changes and population changes allows us to understand how these changes in our application methods are able to impact our safety for our streams and the risks that could occur with them. The forest managers are then able to take this information and decide where, when, and how to apply the pesticide to make sure that they are minimizing risks while still protecting against these invasive species. Ultimately, my research is about balance. One part of the ecosystem shouldn't come at the cost of another. So the next time you take a deep breath, think of Leonard and the small things that protect our forests and streams. Thank you. Okay.
Awesome. Thank you so much, Hadley, for reminding us of that balance and for all of us to think about the impact and thinking about when we take that deep breath. All right? So bring those phones out. We have our next question coming up. So what was Hadley's response to if your research had a warning label, what would it say and why? Our friends at home are already ahead of the game. So the three options, if you can't see it maybe, caution. Results may vary depending on rainfall, slope, and your sense of humor. The middle option, warning, could alter your understanding of where clean water truly begins. And the last option on the far right, side effects may include mud, back problems, and minor existential dread. And our students, it's coming to be finals week, and some of them are feeling that, so we know that. But they are crushing it, and I know some of them are presenters downstairs talking about some of their finals tomorrow. So after this, they're going to go and study like all of our great students do at the University of Georgia. It's going to give you all a few more seconds. It seems like we're really feeling the side effects may include mud, back problems, and minor existential dread. I know, I do too. Giving you all a few more seconds to fill out those answers there. I guess we're not really feeling the caution. The results may vary depending on rainfall slope and your sense of humor. Okay, that middle option is really getting some traction there. But if you can't see, we have about 156 people joining us, so both here and online. So we're really excited to see all the supporters in the room and virtually supporting our student presenters today. Again, remember to take a few notes on what Hadley spoke about, whatever spoke to you, write that on your card, so that way we can think about that as we go into our People's Choice voting at the end of all of our presenters. Okay, a few more middle options there. But I think the majority got it right. The answer is side effects may include mud, back problems, and minor existential dread. All right, well, we are going into our next presenter. We have Oscar Sedegat, who's studying poultry science. His presentation is entitled The Tale of Laying Hens, Told by Immune Markers.
Imagine running a marathon every single day. That's what it's like to be a laying hen. Her body is pushed to produce an egg almost daily. Now add a broken bone in your chest wall. That's the reality for millions of birds. They suffer keel bone or breastbone fractures. But here's the problem. Hens don't cry.
Limp. Hens don't cry. They don't limp. They don't hold up a little sign that says, I'm in pain. From the outside, they look perfectly fine. And yet inside, they may be silently suffering. So, how do we give a voice to the voiceless? That's the question at the heart of my research. And my answer is the immune system. Think of the immune system like a diary that the body keeps. It records stress, pain, and injury. Every experience leaves an entry. Certain cells rise, others fall, signals change, almost like whispers from the hen's body. My job is to read that diary. In my study, I'm comparing immune markers in hens with breastbone fractures to those without. If we can find consistent patterns, those markers become a translator. They tell us, yes, this hen is hurting. Or no, she's coping. And why does it matter? Because welfare isn't just a kindness, it's connected to everything. Health, sustainability, even the eggs on our breakfast table. A hen in pain lays fewer eggs, eats less, and lives a poorer quality of life. And when you multiply that by the billions of hens producing billions of eggs every year, you will start to see how small improvements in welfare could have a massive impact on animals, on farmers, and on all of us who depend on them. So here's the big picture. If we can read the story hens are already telling through their immune system, we can design better housing, improve management, and breed for stronger, healthier hens. In short, my research is about giving hens a voice, not through words, but through biology. Not through sound, but through science. Because when we listen to their story, we can change it for the better. Thank you.
Awesome job. Get up for Oscar. he did a great job, and we're flexible, and even with the technological challenges, he still did great. So we have our next question. Which fictional character from a book, movie, or show would make the best collaborator for your project, and why? Dr. Dolittle, Foghorn Leghorn, and Yogi Bear. I see some Gen Z folks in the room. If you don't know who these people are, turn to the millennial or Gen Xer next to you, and they can explain to you who these are. Wow, thank you for laughing. That means a lot. But these are really great shows and movies to watch. If you haven't watched it, you definitely should. Okay, we're thinking Dr. Dolittle. I'm surprised the lack of Foghorn Leghorn. Chickens, hens, no connection there? Okay. I know folks at home are also engaging. We appreciate it. All right. I think, maybe we're thinking more Foghorn Leghorn. I do not want to bias the vote. Sorry if I am doing that. But if you're feeling Dr. Dolittle, make sure to do that as well. Okay, is everyone putting their votes in? We're putting this on our to-watch shows later if we need to. All right. I think we're seeing a winner here. Yep. And so Oscar said Dr. Dolittle, finally someone who could just ask the hens how they're feeling. Go to, right? Obvious answer there. And so, as I said, we are having each presenter be considered for the People's Choice. So Oscar had a great presentation context, and so make sure you take notes down on your sheets, and remember that later for our People's Choice voting at the end of all of our presenters. So up next, our next presenter, Vijay Mohakar. He is studying textile, merchandising interiors, polymer, fibers, and textile sciences. His presentation, From Nature to Knee, a New Era of Visco-Supplementation.
Have you ever heard the phrase, a well-oiled machine? It describes something running smoothly, effortlessly, like car, bike, or plane. Now imagine your body in the same way, especially your knee. Your joints are the gears and your bones are the moving parts. And the synovial fluid in between there is the oil that keeps it gliding. But what happens when this part start grinding on each other instead of gliding? That is what osteoarthritis is. In osteoarthritis, your natural joint lubricant, synovial fluid, loses its viscosity. Its purpose is to have like the cushioning and elastic property, but over time it becomes thin and weak. And this is what causes like a stiffness and the pain in the joint. And this is where the viscosupplementation comes in. And hyaluronic acid injection that restored the lubrication. And if you remember the Tin Man from the Wizard of Oz, he didn't need a new joint. He just needed a little bit of oil to move again. And hyaluronic acid injection is expensive, is more short-lasting, and it takes like a high amount of time to make it. So I asked, can I make this joint lubricant from something which lasts longer, is sustainable, and it's easy to access? The answer is yes. I'm developing a viscosupplementation from nature-based product, from plants, extracting the cellulose and into its purest form, and adding it with the natural polymer together and making a gel out of it. This gel mimics the same natural lubricant properties of the synovial fluid, like lubrication, cushioning, and shock absorption properties, while retaining its long-lasting biocompatibility. When we think of the osteoarthritis, it affects 32 million people in the US alone, including athletes, grandparents, and the people who just want to walk without pain. When we restore lubrication, we just don't fix a joint. We restore movement. We restore independence. We restore a quality of life. And such that a well-oiled machine isn't just a phrase, it's a science. Thank you.
Awesome job, Ajay. And so let's bring those phones out. Again, our people at home are well ahead of us. And so his question is, if your research were an animal, what would it be and what would it be doing and why? So your options: a mountain goat climbing a steep terrain, a woodpecker hammering on a tree, and a spider spinning silk. Okay, we have kind of two options in the lead here, our mountain goat and our spider spinning silk. Some thoughts? We're just happy there is no longhorn as an option for Saturday, right, Boo? We saw some of them in town this week already. Okay, mountain goat climbing a steep terrain. Are there other votes coming? And I will say, Vijay mentioned Wizard of Oz, and so that was not a plug for the new Wicked movie coming up, but maybe it is, who knows? He is putting all the pop references in his presentation, so really great. Okay, I think we have a lead here, a top winner. A few more votes. Let's see, anyone else want to get those last votes in? But also make sure you're writing those notes down on Vijay's presentation to be considered for your People's Choice Award at the end of the presentation. Okay, I think we can try to see what those results are, maybe. Okay, the majority, you were wrong. Sorry about that. Just give yourself a pat on the back. But the answer was a spider spinning silk, quietly creating something incredibly strong, lightweight, precise, and full of potential. Makes you think. So our next presenter is Allison Arney, who is in the College of Veterinary Medicine, specifically Comparative Biomedical Sciences. Her presentation is entitled Cats and Kidney Disease.
Let's talk about my favorite organ, the kidneys. While they may not be as glamorous and as famous as the heart and the brain, they're arguably just as important. Your kidneys are a paired organ located in your stomach, and their primary goal is to filter your blood, meaning they take your blood filled with trash and gunk and toxins and turn it into clean, healthy blood and a waste product, your urine. Your kidneys are also responsible for things like your hydration, your blood pressure, and so much more. If I haven't convinced you yet that they're wildly important, I'll tell you that your kidneys filter your entire bloodstream in just 30 minutes. So, everyone, please say a huge thank you to your kidneys every 30 minutes for saving your life. Now, what if I told you that you've lost 75% of the function of your kidneys? And there's nothing you can do to gain back the function you've lost or prevent further loss from occurring. That's the unfortunate reality for many cats in veterinary clinics. 8 in 10 cats over the age of 15 have chronic kidney disease, which is a progressive decline of kidney function over time. And we have 2 problems with kidney disease. The first is that we diagnose it too late because our tests only flag as abnormal when 75% of function is gone. The second is that we don't have appropriate treatments. Our treatments are only symptomatic, meaning they treat the signs of disease, but not the disease itself. So, your cat is dehydrated, I can give it more water. Your cat is nauseous, I can give it anti-vomiting medication. There is nothing for me to give for the kidneys. These 2 things make chronic kidney disease a leading cause of poor quality of life and death in cats. So this is where my project comes into play. I am identifying molecules that can be used to diagnose kidney disease earlier and to treat it at its source. By taking the blood and the urine of 50 cats of varying stages of health and kidney disease, I can run biological tests to identify these molecules. By looking at the molecules that are different between health and disease, I'm able to pinpoint which ones can be used to create a test or to be used to create a treatment. My goal is that cats will live happier, healthier, and longer lives with this research. I know that we love our furry friends, So let's keep them around for longer. Thank you.
That's right. Give it up for Allison. That's awesome. And so bring those phones out. We have another question for you. Her question is, what is one word that sums up your research journey so far and why? So the options are complicated, transformative, resilient, magnificent. And so we're kind of changing it up with the visuals. It seems like we're thinking a lot of resiliency. That's awesome. I know some people may have thought of their cat at home. Okay, transformative, taking the lead. Get those answers in. And while you are doing that, if you already answered, make sure, again, you take those thoughts you had from Allison's presentation, put those on your paper and your phone as you begin to vote for People's Choice. We're already halfway through. Our presenters have been awesome. Let's give it up for them. Okay, we're still thinking transformative, maybe a few more resilient, complicated, kind of neck and neck, and then magnificent. No one's really thinking that. Remember, what is one word that sums up your research journey so far and why? All right, I think we can maybe look at that answer. And she said complicated. So hey, we're thinking positive for Allison. So she said complicated since she is looking to discover and classify new testing and treatment target. My research quickly can get complicated with hundreds of biological pathways and molecules. So that makes sense why she thinks it's complicated. So we are now going to look into our next presenter. Give it a few more moments. I'll let you read through that. All right, so our sixth presenter tonight is Kendall Clay. Kendall is studying cellular biology and neuroscience. Her presentation is called That's Dope, Uncovering the Mystery of Dopamine Neuron Regulation in Planarians.
I would like for everyone to take a moment and think. Do you know someone affected by a neurological condition like Parkinson's or Alzheimer's? Do you have a loved one that suffered from a traumatic brain injury or a stroke? All of these neurological conditions have one thing in common. The patient's neurons accumulate damage and then they die. And this is such a critical problem for human health, yet we don't have any solutions to regrow these lost neurons. So how do we go about tackling this problem? Well, let's look at nature. Nature has already solved the problem of regeneration really well in the animal kingdom. And for that, we can look to the planarian. This planarian, the aquatic flatworm, is capable of whole body regeneration. In just 7 days, it regrows a whole new head, complete with a new brain. That's amazing. And this brain is full of many different neuron types, all in the right ratios and precise locations. How does this worm do this so impressively? Well, we know that one aspect of this process is due to the population of pluripotent stem cells. And these stem cells are able to give rise both to more of themselves and also to make all of the mature cell types in the planarian body. But we don't know what genes the planarian stem cells use to make new neurons. So to answer this question, I focus on one neuron type, dopamine. Now, other than them being dope, you might be thinking, why would you even want to focus on dopamine neurons? Well, they're implicated in movement as well as in Parkinson's disease, they're lost in humans. So in our planarians, I have a lovely marker of dopamine neurons shown up at the top, and the brain is full of these neurons. However, when I get rid of the genes LMO3 or APP, you get about half as many of these neurons. And I also found that these two genes are expressed in stem cells that will then become dopamine neurons. So that led me to conclude through my research that LMO3 and APP, these genes are crucial for the regeneration of dopamine neurons in planarians. Now, you're like, oh, well, is this just a worm fact? No, this actually might be important for human biology as well. These genes, LMO3 and APP, are expressed in human dopamine neurons, yet no one has ever used them in functional trials for therapies for Parkinson's. So maybe our ability to learn these things from these worms and include these genes might be able to benefit human therapies and hopefully Through the inclusion of these genes, we can convert patients' stem cells into dopamine neurons readily and effectively, and hopefully the insights from our humble worm will power the next generation of regenerative therapies in human patients. Thank you.
Awesome job, Kendall. So I think we all can say that was dope, right? I think some of the people are bringing that back into the vernacular. So let's bring our phones out. And so the question was, if your lab or project group were a band, what would your role be and why? Would Kendall be the lead singer, the producer, the writer and keyboardist, or the drummer? Okay, we're thinking the producer in the background, mixing things together. Love it. Writer and keyboardist. Lead singer, drummer. I think they were giving Kendall a lot of great compliments. That's awesome. Okay, the producer, we're really feeling that one. Kendall, you may have a future there as well, along with your neuroscience plans in the future. Okay, so everyone put those votes in. Like I said, also make sure, collect those thoughts that you had from her presentation and put those on the paper or in your phone as we near the end of the presenter soon. That was our 6th presenter, so they're doing great, and we're really excited to hear our last 4. But let's see what the answer is. Okay, writer and keyboardist. Maybe she's gonna do all of them. We'll see her future there, the drummer, the lead singer, but we'll give her a compliment. We think she's gonna be the producer in the background. So here is why Kendall said the writer and keyboardist. The worms are doing all the singing. I'm just going along with their music. It's my job to think of questions and experiments, also known as the lyrics, but I'm not deciding the results. I'm just harmonizing along and laying the accompaniment so the results can really shine. That's a pretty good answer. I understand now why she's the writer and the keyboardist. And so our next presenter is Margaret Lemons, who is studying comparative biomedical sciences in large animal medicine. Her presentation is entitled, Unraveling the Sticky Web, How the Body's Defense Can Go Wrong in Babies.
Imagine your body has tiny superheroes in the bloodstream. First responders on call, ready to fight infection. These heroes are your neutrophils, a type of immune cell, and the villains, bacteria and infections trying to invade. When the villains invade, the consequences can be devastating, especially in newborns. Their bodies can't control the fight against infection, and their immune response spirals out of control, damaging their own organs. That condition is called sepsis, and it is a leading cause of death among human and animal babies worldwide. But neutrophils have a secret weapon. When they sense danger, they do something extraordinary. They shoot out sticky webs, like Spider-Man. These webs of DNA and proteins trap and kill bacteria before infection spreads. Scientists call them nets, neutrophil extracellular traps. When nets work properly, they save lives. But just like Spider-Man's webs in the movies, they don't always land where they should, and that can cause collateral damage. In the body, it's the same story. If neutrophils release these webs too late, infection spreads. If they release too many, webs tangle healthy tissue, block blood flow, and organs fail. Here's a devastating twist. In newborns, neutrophils don't make these webs properly. Once infection takes hold, their immature immune systems become quickly overwhelmed. We believe this is why babies and foals, or baby horses, are so vulnerable to sepsis. Both share the same life-threatening weakness, neutrophils that can't mount this Spider-Man defense the way it should. When sepsis strikes, the scene is heartbreaking. A premature baby and foal, both in intensive care on fluids, oxygen, feeding tubes, fighting to keep their organs alive while their immature immune systems fail them. It's a compelling reminder. These are not just data points. They are real patients whose lives are on the line, and understanding their immune response could mean the difference between life and death. This is where the foal brings us hope. Because their immune system develops like ours, they are a powerful model for studying sepsis. By learning why foal neutrophils don't make nets properly and how to fix it, we may uncover answers that could transform care for both human babies and foals. My research has shown that adult horses and foals make nets differently. But the question remains in foals. What is happening in those first few days of life when they are at greatest risk of sepsis? And why doesn't their Spider-Man response kick in the way it should? In the end, this work is about these patients. By cracking the code of nets, these Spider-Man webs of the immune system could save lives everywhere.
And give it up for Margaret. Awesome job. And so pull those phones out. We have our next question coming up. And so her question was, what emoji best captures your research journey so far and why? I'm going to do my best to explain these. So the little grimace face emoji. We have our microscope emoji, a roller coaster emoji, and then a spider web. Okay, we're really thinking spider web. That's awesome. That's the lead. A few grimacing face, a few microscopes, a roller coaster. I know we're all thinking about the connections now between foals, horses, and babies. I know all the moms here are thinking when you were pregnant, your child was definitely a horse running around in your belly. I'm sure you felt that. All right, we're thinking spider web. We want to give a moment for our folks at home to also vote. But as always, you all know the drill. Make sure you collect those thoughts from Margaret's presentation. Write those down as you consider your People's Choice Award and a few other presentations. So we're super excited to see that in a little bit. But, okay, really between spiderweb and maybe roller coaster. Let's see that answer. Okay, yes. Oh, I heard that. Yes, you're playing along at home. That's great. So the spiderweb, and here's why. Because her research is all about sticky webs, as you all heard. You were listening. Good job. Both literally and figuratively. Neutrophils shoot DNA webs to trap bacteria, and her research journey has felt like untangling one scientific web after another. So we get that. So that's why Margaret chose that spider web there. So let's give it up for Margaret one more time. Okay, if you're counting down, if you're following along on your sheet, we have 3 more presenters. Our next presenter is Adhar Yegane, who is studying chemistry, specifically materials chemistry and nanoscience. Her presentation is entitled Shedding Light on Hidden Dangers in Our Homes.
Today, I want to talk about your everyday companions. Just take a moment and think about the fabrics that are in direct contact with your skin all day. But if I told you all these textiles surrounding all of us could be a potential danger. Yes, I'm talking about counterfeit textiles. These are fake fabrics produced illegally but sold under familiar, trusted names. Shockingly, 60% of counterfeit goods seized at our borders are textiles. That means the clothes that we wear, the shoes on our feet, and the upholstery in our furniture are the main targets of counterfeiting. These goods come from illegal sources. They may contain toxic chemicals, and they may not perform as promised. Imagine buying a curtain labeled flame resistant, but when a spark hits, it goes up in flames. See, it's not just about being fake. These counterfeit textiles can put our safety at risk, and that's why we step in. To make textiles harder to counterfeit, we developed an invisible security pattern that these counterfeiters cannot even see, let alone copy. To have this invisible pattern, we explored a very special pigment. To the naked eye, it looks like an ordinary pigment. But under near-infrared light, it reveals a hidden glow, which is visible only under a special camera. We were able to produce that pigment fast, cost-effective, with a strong glowing property. But there's another challenge. How can we put that amazing pigment into textiles? To solve that problem, we took inspiration from nature. Just like a spider spins the web to trap its prey, we developed a similar web-like structure, but in nanoscale. It traps the pigment and keeps it on fabric strongly so it doesn't wash out, even after going through your washing machines. we were able to produce a formula that can be printed easily on any type of fabric. Look at this fabric. Can you see anything on it? Exactly, it looks completely blank, right? But under near-infrared light, ta-da, the Georgia Bulldogs logo is shining, like always. This property make it almost impossible for counterfeiters to fake, and the customs officers at borders can use portable infrared scanners to verify the secure pattern from the producers instantly. We strongly believe this work can help you to purchase a product which is authentic, safe, and trustworthy. Thank you for joining me on this journey toward a safer future for all of us.
That's right. Give it up for Adhar. And so pull those phones out. We have the next question. Bringing some memes in here. So she was asked, what meme best sums up your research or grad school experience? And so kind of like with the cartoon for the maybe younger generations, if you want to explain what a meme is to the other folks in the room maybe, it's write a picture that's kind of be funny, pokey. I saw someone in the group doing this, 6'7". I think that's not too much of a reference yet for some of us, but some people do that 6'7" stuff still. But here are some of the answers here. Don't ask. It's a picture of a horse. It says, don't ask, but yes, I could use some help. The second option is me trying to explain my PhD research to family members I see once a year. Hey, Thanksgiving is coming up, maybe Christmas, Hanukkah, whatever you celebrate. Having to come up with that for our graduate students is always a big task. The last one is, this is fine, everything's on fire. You may feel that way, but the weekend is almost coming up. We're almost there. So put your responses in, everyone. We're really thinking that this is fine, everything's on fire, but it's fine. It's fine. I was just thinking while Adhar was presenting, those were pretty cool shirts. Maybe our next night game in Sanford Stadium, get a few couple thousands of those. That'd be pretty cool. Okay, we're really thinking communication chaos or cool under fire. I'm sure some of us can feel like that middle meme in some of our meetings, trying to diagram some themes, arguing a nice, healthy debate. Okay, but we're really feeling this is fine. All right, let's see that answer. Oh, hey, but 37 of you got it right. So here is the reason. I think it's pretty obvious. That's how she felt sometimes, explaining it to her family. So that's pretty cool. Really embodying the emotion of going home to a holiday and people asking, what do you do? And they're like, now they can do it in 3 minutes, but usually it takes about an hour, right? So give it up to Adhar. So up next, we have Anshul, who is studying crop and soil sciences. Her presentation is entitled Peanut, the Uncrushed Oil Potential.
Please raise your hand if you buy peanut butter frequently. Whoa, plenty of hands. Now, keep your hand raised if you buy peanut oil just as often. No hands? Peanut oil in the US is twice as expensive as peanut butter because despite being the 4th largest peanut producer in the world, US imports 26% of peanut oil. Surprising, isn't it? That's because 60% of peanuts in the US are used for food products. Now, what might look like a successful industry is just an illusion. Every year, 10% of edible peanuts are trashed just because they do not meet the beauty standards of food industry. These losses skyrocket when drought and aflatoxin, a cancer-causing contaminant, hit the fields. This happened in 2019, resulting in a huge loss of $125 million. The question is how to strengthen the US peanut industry against these environmental and economic challenges. I have a promising solution: diversification to oil production. Peanut seeds contain 44% oil, and we are certainly overlooking that potential. Here's the game changer. Food scientists have discovered that peanut seeds, irrespective of their appearance, and even the ones with aflatoxin, can be used safely for oil because aflatoxin is removed during oil extraction. I know. You might be wondering, why isn't the US growing peanuts for oil then? Well, there is another problem. So far, peanut farming in the US is designed only for edible crop. So, farmers do not have a roadmap to grow a successful oil crop. Therefore, I am developing a production strategy to help farmers grow peanuts commercially for oil. Now, there's another problem. It is how to grow it actually in the field. For that, I have planted different combinations of peanut variety, seed rates, and row patterns in the seed, and I collect seed samples and analyze the seed oil percentage. By comparing the results across these combinations, I am determined to find the best peanut variety, seed rate, and row pattern that gives the consistently higher oil yields. The farmers can then use this strategy to grow a successful oil crop of their own. Isn't that amazing? With my research, I hope to reshape the peanut industry in the US towards a more profitable future. Thank you.
That's right. Give it up for Anshul. Okay, bring those phones out for our questionnaire. So she was asked, if your research could change one thing about the future, what would it be? Your answer options are teach peanuts to harvest and press their own oil, total farm efficiency. The second option, making farming more resilient and rewarding for farmers. And the last option, replace all the other crops with peanuts because they're clearly superior. And I know we feel that way, especially here in Georgia. Put those answers in. And we can't mention peanut butter without asking the age-old question, right? So we're going to ask you crunchy or smooth. Or creamy, sorry. Smooth. You know my answer already. So who thinks crunchy? Raise your hand. Okay, we see some crunchy folks. Who thinks creamy? You may be the best sample size. I'm really seeing 50-50 here. So we cannot bring our insights to any peanut butter producers, but hey, it's good. They all belong in our pantries, right? So we're really thinking making farming more resilient and rewarding for farmers. I really like the first one, total farm efficiency. We love that. Okay, any other options? People at home, make sure you're voting. Okay, oh, another vote for replace all other crops with peanuts because they're clearly superior. Okay, any other votes coming in? All right, let's see that answer. And the majority was right. So she said making farming more resilient and rewarding for farmers, and here's why. I want to help farmers produce high-quality crops like peanuts in ways that use resources wisely and adapt to changing climates. And so up next, we have our last presenter, Adi Mavengere, is studying workforce education and instructional technology, specifically learning, design, and technology. His presentation, attention, listen to what the eyes say.
Indulge me for a moment. If you're a student and you've used Zoom or a similar platform for a class, please raise your hand. If you're not raising your hand, please take a moment to look around at someone who's raising their hand. Thank you. Please lower your hands. Now, if you're raising your hand, you are one of millions of students across the world who have possibly missed an opportunity to get full support from an instructor due to a limitation in video conferencing platforms. You may agree with me that increasingly human interaction is occurring through platforms such as Zoom. And when such interactions involve instruction, we term that screen-mediated learning. Now, the limitation of video conferencing platforms and the challenge that we have with screen-mediated learning is that nonverbal communication is obscured. 70% of human interaction is nonverbal. So when it's obscured, it becomes very difficult for instructors to be able to gauge whether students are actually learning anything. In the absence of any other nonverbal communication, let's listen to what the eyes have to say. My research focuses on understanding the relationship between attention and oculesics. Oculesics relates to eye movements, pupil behavior, and eye-related nonverbal communication. Research has it that oculesis is a good indicator of task difficulty, and it is a good indicator of when students are distracted or disoriented. By taking oculesics and applying it to screen-mediated learning environments, instructors are better able to determine students' attentiveness. In this way, instructors are better able to support students. Through oculesis, students are able to get the support that they actually need, and that improves learning outcomes. Through oculesics, in screen-mediated learning environments, we are able to improve the learning outcomes for the people who raise their hands and millions of other learners across the world. By decoding the language of the eyes, oculesis is improving learning outcomes. Thank you.
That's right, give it up for Adi. Okay, our last polling question of the night for now. So, the question is, if your research were a movie, what would the title or genre be and why? Okay, we have some great responses up here, a lot of moving options. The first one, science fiction, the language of the eyes. Historical fictions, eyes in the night. The third option, crime thriller, the secret of their eyes. And the last one, drama, the look of silence. These are pretty good titles. I think regardless, they should all be submitted maybe for an Oscar, right? And we can bring our, I think it was Kendall as our producer to make the movie. Okay, we're really thinking drama, the look of silence, maybe crime thriller, the secret of their eyes. No one's feeling historical fiction. I think they deserve some love a little bit. Historical fiction, eyes in the night. I'm sure there are some historical fiction buffs in the room. Okay, woo, yeah, woo. Okay, a lot of science fiction, the language of the eyes. Any other thoughts? Remember, Adi was our last presenter, so collect your people's choice thoughts. Get together, maybe look at the back of your agenda there and see all 10 options, because we're gonna use that information soon. Okay, the science fiction, language of the eyes is still leading. Get those votes in, those at home. Okay, let's see that answer. Okay, you all got it. Science fiction, the language of the eyes. And here's the reason why. It would be a sci-fi. The movie would be futuristic about how people communicate in a world where verbal communication gives way to the language of the eyes. Everyone, let's give it up for your Fall 2025 finalists. So they all can hear you. They're in the crowd. They're back here. They've done awesome. But thank you all for your participation and engagement. I hope you have learned a little something tonight, but we're not done. We're actually going to have the judges deliberate. They can head out while you all vote for the People's Choice Award. I've been mentioning it all night. Look through your notes. We have this QR code on the screen for you to vote for People's Choice. People at home as well. For the QR code, open your camera app. Scan that QR code. It should bring you to a link, or open a web browser of choice on your phone. Go to menti.com, type in that code. You can type in, choose your presenter for your People's Choice Award. Remember, that is one of our 3 awards there. So you will submit your vote through the same process if you answer the questions tonight. The code's on the screen. And for those viewing virtually, the link and code will be shared in the comments section on Facebook. And so while our judges are deliberating, we have a treat for you all, another graduate school student. So here's some background. November is Spotlight on the Arts Month here at UGA, a time when creativity takes center stage across campus. From galleries to theaters to concert halls, the arts remind us how powerful storytelling can be. Tonight, our finalists took stage to do something similar. They've blended science and art, crafting performances that distill years of research into just 3 minutes, within the stringent parameters of only speech and a static visual slide. It's research, but it's also rhythmic, voice, timing, and imagination. So, in the spirit of Spotlight on the Arts, we have a presentation for your enjoyment that extends beyond the parameters of the formal 3-minute thesis competition. Please welcome to the stage Jennifer Lee Whitehead.
When my grandfather passed away, and I promise this isn't too sad of a story, I was about to go to college. I was living in Cincinnati, where I grew up, and he lived just across the river in Maysville, Kentucky. And he had this little house with a big field out back, and in the front there were train tracks, and then you crossed the train tracks and you were right at the river. And I remember growing up, I would go and I would practice my gymnastics routines on the cinder blocks lined up out back. I would steer the tractor when he let me. I would pick all of the fresh tomatoes out of his garden. And the day that he died, it was a late July, gloomy, muggy day. It's hot, it's humid. I know we get that in Georgia. And as we're driving down to visit him, the storm rolls in. And it comes out of nowhere, and it starts coming down hard. We can't see anything. We're driving as safe as we can, but it's just pouring down the road. And as we're driving, the largest lightning strike that I've ever seen moves across the sky. It lights it up like it's the middle of the day. And the thunder that came after, you felt it more than you heard it. And something about my grandfather, he was a simple man. He knew what he liked, right? He was a kind man, he was involved in his community, and he liked the simple things. And one of his favorite things in the world was to crawl under the covers and take the world's best nap every time it stormed. So as we're driving down, we get to his house, and he's already gone. And when we talk to my uncle, he says, the time that he left us was that exact moment that lightning struck, that lit up the sky. And when we walk in to the bedroom, where he's snuggled up in bed, looking like he's taken just the best nap, I'll never forget, the radio was playing. And on the radio, this world is not my home, I'm just passing through. My treasures are laid up somewhere beyond the blue. The angels beckon me through heaven's open door, and I can't feel at home in this world anymore. Hi, everyone. My name is Jennifer Lee Whitehead. Thank you for the lovely introduction before. I am a first-year PhD student in the Department of Theater and Film here at UGA, and I firmly believe that art has the ability to elucidate things that are otherwise unknowable. Art takes the things that are balled up in your chest and caught in your throat, and they become understandable. My goal when I'm creating art is to take something that is a feeling that is so visceral and true and purposeful and express it in a way that makes you feel understood. And I think that art is uniquely positioned to do that. And my process when I'm creating a work like this piece, Do Not Say Farewell. We've got a couple of excerpts from a larger piece that I've done previously. And my process, I'm sure, is not too dissimilar from these folks who've just presented all of their research, right? I write and I write and I write, and I figure out, is this the best way to communicate what I'm trying to communicate? And is this clear? Is it understandable? And I take that time and I write and I write and I write, and then I throw out all of that, and then I write and I write and I write. And sometimes I do all of that, and then I still start over the night before, not speaking from experience. The thing about performance, especially performing from personal experience, is that a lot of times it feels like you're ripping your chest open, and you're tearing out your heart, and you're holding it out to a pack of ravenous dogs, and you're saying, I really hope this tastes good. And I think that that's a lot like what all of these competitors did this evening, because they took their passion, their research effort, so much effort, all of the incredible ideas that you know they have in their head, and they said, how do I communicate that in 3 minutes? Now, to finish this off, we're going to do some experiential learning, so I hope we're all excited for that. I'm going to separate us into, sorry, it's hard to see you guys out here. I'm thinking we'll just go right in half, so 1 and 2. So group 1, yeah, 1 over here. So let me just establish some rules first. When I go like this, everyone can stop. And that's all the rules. So, group 1, I'm going to have you go, sorry to the sound guys, I didn't practice this. We're going to go shh, 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 shh. Stunning. Okay, group 2, you're going to go shh, do not say farewell. Do not say farewell. Do not say farewell, my love. Do not say farewell. Your farewell to me is like the sun. Yes, it shall dip beneath the horizon. Yes, it shall be dark. Yet there is a moon to shine light while I wait. There is day that will come again. Wherever your farewell might be, however your farewell might taste, it will not last. Day will come, life will come, light will come again. I can hold this hope, though I wish you not to go. Do not say farewell. But if you must, say farewell with sunrise in your mind. Say farewell like the sun. And I will look for you in the moon. Thank you.
Thank you so much to Jennifer Lee Whitehead. That was incredible. And so again, November is Spotlight of the Arts Month here at UGA. I'm sure celebrated elsewhere. So make sure you're also, we had some great presenters from various disciplines on campus, from campus today out of our 10 presenters, but also explore some of the incredible students who also are in the arts at the University of Georgia through our Lamar Dodd School of Art, Hugh Hodgson School of Music, some incredible opportunities there. And so, if you haven't voted for People's Choice, now is the time to do so. Make sure you're doing it. We saw the voting back screen. There's a lot of options back there, a lot of votes coming in. People at home, you still have time as well. A lot of votes. A lot of votes. So, I'm going to say, okay. We're going to give it a few more seconds just to allow you all to get those votes in for People's Choice. I know our judges have returned, so that means we know our winners as well. So excited to announce those in a few minutes, but we want to make sure everyone has time, especially those at home, have time to vote for People's Choice. But can we give another round of applause for our 10 presenters tonight, please? Okay. But before we get to our 10 presenters and their awards, we do want to acknowledge our alternates. We have 3 amazing alternates that also did a fantastic job and went through the prelims. We have a couple of them in the audience tonight. If you're able and willing, would you please stand, our alternates who also did a great job with the presentation. Awesome, we have 2 of them right here. Let's give them a round of applause. We want to give them a shout out. These are just 10 and 2 of the amazing graduate students at the University of Georgia who went through this process. I know it was a challenging process, and to really make it both in those positions and these is really a testament to your passion and to all the hard work you've put into your careers thus far. And we know you will go far wherever you go after this step. And so let me make sure okay. So we have the results. I'd like to invite Ron Walcott, Vice Provost for Graduate Education and Dean of the Graduate School, to the stage to help us recognize our finalists tonight. And so we will go through our finalists in presentation order. And so finalists, as I call your name, please come to the stage to receive your certificate from Dean Walcott. As you come to the stage, you will kind of stand under the screen starting on this side and over. And so, first, we have Cameron Wysocki. Give it up for Cameron. Give it up for Hadley Copeland. Next up, Oscar Sedegat. Give it up for Vijay Mohakar. Next up, Allison Arney. Our next presenter, Kendall Clay. Give it up for Margaret Lemons. Next, we have Adhar Yegane. Next, Anshul. And our last presenter, Adi Mevengare. And so here are your 10 presenters. Let's give it up for them. Yeah. We know they've put in a lot of work to get to this point, and so we just want to make sure that they all know we appreciate their hard work and all the work they've put into this event. And so the one we've been waiting for, we are now going to announce our winners for the evening. And so we're going to receive our cards. Thank you so much. And so we'll begin with People's Choice Awards. Tonight's winner is Hadley Copeland. Her presentation was entitled Risk Runs Downstream, Managing Forested Watersheds. Congratulations, Hadley. Next up, we have our runner-up in the 2025 Fall 3MT competition is Askar Sedegat. The presentation entitled The Tale of Laying Hens, Told by Immune Markers. Congratulations, Oscar. And for our winner, Anshul, presentation entitled Peanuts, the Uncrushed Oil of Potential. Let's give it up for our winners and all of our presenters. And so for everyone in the audience, now's your time. Take your pictures of our 10 presenters. But we want to thank so much to all of our wonderful competitors, their academic programs, the staff that made this event possible, and to you for joining us tonight, both in person and at home. We hope you had a wonderful time and that you will stay in touch with the graduate school on social media to learn about future events and ways to support the work of 9,000 plus graduate students at UGA. We'll see you next year. All right. Yeah, have a good night, everyone.